think we'll give a 30 more seconds, let everybody roll in, then we'll get started here. I'll just do some quick intros while we're waiting. I'm Neil McCormick. I'm the Global Product Development Manager for Adjuvants and Biologicals Crop Nutrition. So I've managed those two lines of business alongside this guy, and I'll let him introduce himself here.
Before I introduce myself, just for questions, answers, we got a little long last time because we were doing it through the session. This is being recorded, so if somebody has a question, we'll save them for the end if possible. And just raise your hand and they'll bring a mic. That way on the video it'll pick up the question. So if we can ask everyone to do that, that would be great. I'm Dean Skinner. I'm an agronomist with FBN and work on the biological side of it and the science on how to use the different products.
Cool. Yeah.
Yeah, I think everyone's kind of in here now. So
I don't know if you want to tell about your life history or anything.
Well, I grew up on a farm in, you got the changer?
Yeah, sorry, I have the screen up here. No, I'm just joking. But so we're going to get into the topics here today. Is going to be yield gain versus yield potential. So we just want to do a quick, there's a lot of buzz out there. There's, 5,000 products, 10 bushels a piece. You have you can, if you bought all the bio products out there, you could have a 5,000 bushel corn crop on, so it's like, okay, let's cut through all that and give you a good sense of what can we actually do, what's the practical agronomic application and use of these products. So we'll go into stress mitigation, soil health, nutrient efficiency, and then the products that we've launched to be able to give you guys some solutions to some of the problems we're going to lay out. So when we look at, that's the picture everybody has. We'll add this, gain 5 bushels. Everybody has their chart, and that's how these products are sold a lot of times. When you look at a chemical, like we don't, the chemical we're trying to solve for biotic stress, so the disease, insects, weeds, fertilizer, we're solving for nutrient supply. So I'm short on a nutrient, I need to add it back in. So what are the actual benefits in this spectrum that we can solve with biology? So there's two things. One is abiotic stress, so non-living stress, so weather, chemical, soil stresses, high salt, pH issues, all those types of things. Then nutrient availability. So once I've added the nutrient, how do I make sure that it stays stored, I don't lose it, it gets to the plant? So I'm investing a lot of money in fertilizer, so how do I make sure that it's actually getting to the plant effectively is what biology can do. So it's really those two things. There's a lot of products out there, but majority of them are going to solve those two issues. So to explain that to you, I'm going to turn it over to the agronomist to go through how that works and how and why it works, and then we'll get into some products.
Yeah, so this slide kind of indicates the yield potential of the plant. So everything that negatively impacts that plant will reduce the yield potential through the growing season. So we just, we think about nutrient supply being one of the big factors, and of course different stressors be they weeds to compete with the crop, or insects or fungus. We can use the chemicals to control that, but we haven't really, people haven't really dealt as much with nutrient availability. How can we get the nutrients out of the soil and make them available to the crop? And then of course just different stresses, how the biology can aid the crop in becoming more resilient. And this can help us to, hold our yields higher, so we, can maintain more of that yield potential.
So we're never really gaining yield, we're just fighting to maintain what we have.
Yeah, trying to keep as much as we can. This is Liebig's law of the minimum. I don't know, some of you might be familiar with that. He was one of the fathers of soil science. And basically he showed that we need all of these factors, and if something's limited, say we only have 60% of a nutrient that we require, that's going to mean that we can only grow about 60% of that crop's yield potential. And otherwise the yield will spill out like the water in the barrel here. Flying in there, you can see what does a plant require for nutrients. And we think about nitrogen, potassium, and phosphorus as our major nutrients because that's probably what we spend the most money on. But really and truly, the plant is made up predominantly of carbon, oxygen, and hydrogen. And we'll talk a little bit more about some of the importance of those minerals as we go through. And you can see all of the different minerals are required within that plant. This kind of just shows us, we think that we've farmed most of the, or mined most of the nutrients out of the soil. But really and truly, this is from Sparks, and it's a textbook that most soil science universities would use, shows that there's actually quite a bit of mineral remaining in the soil. Now, these are averages. Of course, every soil is going to be different if we tested, the total nutrients. But it just to show that what we've really mined out is more the organic matter and that function of the organic matter to help make some of these minerals available. So we're going to talk a little bit about how we can gain some of those functions back for our soil. You see the 70 million pounds of nitrogen over every acre. Sounds like a crazy number, but really our atmosphere is 78% nitrogen. So there is a lot of nitrogen there. It's just not in a plant available form. And we'll talk a little bit about how the microbes can help to fix nitrogen for us. This is just a further sort of, we think about if we don't add phosphorus, we're going to be, stealing it from our grandkids kind of thing. But when we think about a healthy soil, and I know Ward Labs has said they've found on good healthy soil roots down as much as 8 feet. This is just a 3 feet, and you can see there's over 10 generations of phos there. And if you think about 8 feet, that's going to extend it about 2.5 times further. So there's actually quite a bit of minerals in our soil. So we're going to look at some of the ways we can help the crop make those more available. And that is mainly through organic matter. We look at humus as the major sort of constituent within organic matter, the most stable product, and we've kind of knocked this down within our soil since we would have broke them in the pioneer days. The decomposing organic matter, it's still adding function and going to humus, and as it breaks down, we'll be getting more and more nutrients from that to make them available to the soil. And fresh residue, of course, is something we want to cycle those nutrients to keep them available to the crop. And then finally, the living organisms are what brings a lot of the function to the soil in terms of they can work with the plant. We'll talk a little bit more about that, how they can work with the plant to make nutrients more available. And as we kind of churn into the soil, we bring them back into the biology and make it available to our crop. So when we designed a lot of the portfolio of how to use these products, we looked at, kind of how nature is doing this, and we can add living organisms through things like in our Inject product line. Catalyst is more, different food sources for microbes, different signaling compounds to help wake up the native microbes in the soil. And then the Inhabit is humate type products that act like humus and bring a lot of storage and nutrient transfer functions to the soil. A hot topic these days is how do we stabilize our nitrogen and get the most out of it? It's very expensive, and there's many ways it can be lost from our system. When we add fertilizer, it's usually converted to ammonia, and it's a positive or a cation, so it binds to the soil, so it's fairly stable within the soil. And then we have microbes like nitrosomas that actually convert that to nitrite, and then nitrobacter converts it to nitrate, which is really soluble and goes into the water solution. But what can happen if we have, of course, excess water is it can be carried out or leached from the soil, and that's a potential loss for the nitrogen. Or if we have a lot of water pooling and conditions get anaerobic, so there's no oxygen there, some of the microbes will actually take the oxygen off the nitrate molecule, and then the nitrogen is lost to the atmosphere. So to try and prevent this, historically, we've used things like NBPT, which will block the enzyme that converts that, so really just trying to slow this process down. And then, of course, DCD and nitroprion would actually kill nitrobacter. So by doing that and keeping it from, all converting to nitrate very rapidly, when the plant is small, we can eliminate some of those losses historically. But we want to talk a little bit today about how can we stabilize our nitrogen and try and keep it into our system using biology. So again, with the ammonia, and we'll look at how we can fix nitrogen. There's a lot of what would be called nitrogen fixers that can produce ammonia, and that will be converted by the microbes into nitrogen that's available to our crop. Here we see the same steps, so we'll just jump through that about what we just talked about in the previous slide. So what we really want to do with biology is we want to get the plant shooting down the exudate. So the sunlight will come into the leaves, that's light energy, and we'll convert that into what would be a chemical or plant energy, being we're going to take carbon dioxide and water and create sugars, and we're going to create carbohydrates. And those the plant will actually feed into the soil. It'll feed up to 50% of its energy, and that's going to stimulate things like the bacteria to grow. And if you picture bacteria similar to, grass out on the range, we get a lot of grass growing, and then the protozoa is kind of like a cow that comes along and eats that grass. And it's going to have a lot more nitrogen than it needs, so it will excrete a lot of that as sort of metabolites out the back end in the form of essentially amino acids. And the neat thing about an amino acid is it has the nitrogen and it has the energy attached to that nitrogen, but it also will have all the other minerals. The microbes will go grab up all of these different minerals from the soil, and that package is very absorbable by the plant to give a more complete nutrition.
And maybe, Dean, a lot of us are familiar with the Haney, and that's essentially what we're measuring when we use a Haney test, is the amount of nitrogen in that amino acid form.
Yeah, and the carbon form and soluble form, you bet.
Yeah.
So another way we can slow this process down is through humates. They will bind actually ammonia, nitrite, nitrate, and slow that whole process down. And that actually gives the microbes in the plant more time to absorb that, so that we can get it into the plant, and not through the nitrate state, but as an amino acid, which, as we said, is very balanced on the nutrition side. And it actually, because those microbes are replicating themselves, they're very good at going into the soil and pulling nutrients out that would be considered not plant available. And through that cycle, they can be made available to the plant. So some of the ways our products can work with this, we bring a Taurus and Nourish Vitals. They're photosynthetic enhancers. So they will create more of that light energy to form the sugars to build the bacteria, and that system will create more and more energy in our soil. So if we have stress events, the plant actually has a lot of energy to deal with that, and as Neil said, will help to make it more resilient down the road. When we look at catalyst, it is basically, it stimulates the native microbes. It's a probiotic, so we can get a lot of that biology going. The myco is a fungi, very neat fungi, can actually extend much deeper than usually several feet below the root zone. It's able to excrete compounds that are very low pH, very acidic, and dissolve the rock down to about a 2 pH. And that's, if you think about how we make phosphorus fertilizer, we take rock phosphate and add an acid to that to solubilize it so that it becomes plant available. Our Inject N is azospirillum, is a very proven N fixer, as Neil said, helps to fix nitrogen. And we'll talk a little bit more about our Inhabit Boost, which is a humate, and then a starter fertilizer using the boost technology to add it to phosphorus is our Inhabit P. So we want to think about, how did we develop these soils on this planet? And this is sort of a timeline, and it really wasn't until we got into the microbes and the microbes working with the plants that we started to free up and create a lot of these different nutrients. Now, since we started farming and broke up the soils, we've actually lost quite a bit of this carbon. And we want to try and rebuild it very similar to, if you have a house, say, that's been lived in for 50 years, it's kind of broken down. People think about, well, we should sustain that. But really and truly, if we engage the soil building processes, it's like renovating that house and bringing it back to those original conditions and trying to regenerate it. This is just a quick video to kind of help give an illustration of how the life in the soil can really start to break down the trash in the soil. And these organisms, you can kind of see them go down into the soil. And when the plant's there, they like to go up and sort of rest by the roots, because the plant will modulate the pH and make it more comfortable. And they'll excrete a lot of what they've consumed. Again, is a metabolite that will help benefit the plant. And they'll actually dig tunnels from the surface down to the roots, which helps air and water flow into the soil and starts to create some of that structure that we'll look at later with some of our trials, how we've improved that structure. When we think about organic matter, really we have, on Mars, you have all those minerals present, but without that life in the soil, it really isn't going to grow very much. And this is the Osa Peninsula, very rich and diverse, and we've got a lot of different plant and animal species. But when we think we really have a lot of microbes that actually support that, and the nutrient cycle very well, and these organic compounds help create a very healthy soil. But with that, we'll pass it over to Neil to summarize.
Cool. Yeah. So hopefully I gave you guys a good agronomic overview of why do we use these things? What are we actually trying to accomplish? Or what is the potential? So that's our stance. And what you look at conventional crop nutrition, you add fertilizer, replace, add, replace, add, replace, sometimes needing more and more fertilizer as you go down that path. So our goal is really, how do we, we're not advocating you go out and stop fertilizing tomorrow and this is just going to magically grow a crop. It's really add the biology, try to use this process, build your organic matter, and then really, make that expensive investment in the fertilizer stay there, and then also hopefully you can maintain similar yields without having to increase fertilizer. And the potential does increase to decrease fertilizer, but we advocate that's a hard thing to give you a hard fast rule to just go out and cut X amount. It's really dependent on soil tests. Everybody's different. That does exist. It's just it's a long complicated to give you that blanket approach. But overall, we want to help you build that organic matter, increase the nutrient availability, the storage, so make sure everything's that we add stays there, and then water holding, stress mitigation, and then ultimately long-term ROI as we continue to build up the soil. Ultimately, that should show up in ROI, whether it's decreased fertilizer. If I'm building up my water holding capacity and I increase my yield on a really dry year where I would have had a bigger drop, but since I've been building up my water storage, I'm able to maintain my yield better. Those are the types of long-term ROI and investment, and really design the portfolio to where you don't just throw it in, hope you get a gain. We're really helping to give a tool to do some of the things you do with the cover crop, but just try to do it with biological products is a lot of the spirit of what we're trying to do. So getting into the actual products, there's a lot out there for you guys to navigate. What our goal was to really do a good job vetting the technologies out there and land on a really high quality portfolio. So we want to understand, what technologies are out there, how they work, what the mode of action is, make sure it's a consistent formula. So really get all the junk out of the market. Dean and I looked at a lot of stuff, and if you ask, well, what is it and how does it work? If you can't answer that question, we just moved on pretty quick. Because if you don't know what's in your product, it's probably not a good sign, right? So that's the Terry. So we just made sure, and we'll go into that science and agronomy and the products. Convenience, so we just designed the portfolio. Sorry, shock there. But designed the portfolio to be able to sync up with what you're already doing, so we're not asking for extra passes. Go put 20 gallons down, right at the end of specific timing that requires extra passes through the field. We synced it up with all your current practices. We've got a guide that we'll hand out. We'll go through that in about 3 to 5 minutes here, what that looks like. And then data, so we want to generate more and more data to figure out, well, how do we add these products? Is there any trends we can find in how you can reduce fertilizer or by how much? And then just try to get more predictability around performance. And then we also have tools within your account to where if you want to adopt these products and figure them out, you can run on-farm trials and get results back. We have a soil test protocol we developed with Ward Labs we can give you to be able to measure as well. So really the three tenets there.
Before we get into the products, Neil, that was a lot of heavy science.
Yeah, let me back up here. So I think we got to wake everybody up a little bit.
Yeah, wake everybody up a little bit. So let's jump into it.
Right.
Introducing the world's largest straw, capable of sucking nutrients up from deep in your soil. Mycorizol fungi. And to tell you how to get this superstar organism into your field, a fungi himself, Neil McCormick.
Weighing in at 2 ounces, $10 an acre. No, we're good there. So we got their
I
attention.
think that's enough. Woke everybody up.
Yeah, we'll put the dog and pony show to a rest for now and carry on here. So, so have some fun with it. It's a lot of heavy, dense agronomy there, but yeah. So our mycorrhizal, if you guys have used mycorrhizals, there's a lot of powder. It's all spore-based. All the technology today. So this is the only multifunction. 3 modes of action. So it's the only multiple mode of action mycorrhizal on the market. So it's got, in addition to spores, it actually has little cross sections of the roots. So you have the mycorrhizal roots within the product, and then the little hyphae, which are just small subsections of roots. And what that does is it just colonizes, the hyphae are going to colonize a lot faster than the spores alone would. And then it's a gel-based formula, so it's got a 2-year shelf life, so you can carry it over a year if you want to, if you don't use it all. 2 ounces, so super low use rate. Then root growth, nutrient acquisition, water efficiency. So when we think of a cover crop, the bigger root mass we get, the more organic matter that's going to create. So the more we grow so really, hey, how do I grow as big of a root mass in my cash crop as I can to be able to have that stay over through the season to be able to build up soil structure. So that's a lot of the goal there. The second live microbe is the Inject N. Dean, any fancy intros? Are you good? Good, all right. But so this is an Azospirillum brazilense. It's a highly concentrated formula, and Azospirillum is one of the most widely studied and adopted nitrogen fixers. So we just wanted to have a really stable, reliable organism that's had a lot of proof of concept in the field. So it's one, I think in Brazil, it's something like 40 to 50% of the acres have this organism on it. So it's a very highly adopted product there. Goes down at 3 ounces, and then what that does is when you went through the nitrogen fixation, that'll just take the nitrogen gas and convert it into ammonia. Is the main function of that organism. And it doesn't require a root host, so you can you want to avoid using it with bifenthrin, so it's not going to go well with insecticides. It's something you can throw, like high salt fertilizers are probably something to steer away from, so you do want to avoid those, but it's something if you're wanting to get some supplementary nitrogen production. It's similar to what we do in pulse crops or soybean with the nitrogen there, and then just those are the 2 benefits. Next product here is going to be Inhabit Boost. So if you guys have used Humates, Humates are just a couple million-year-old. Started out as a swamp, became a peat bog, and then as it degrades more, it's really the sweet spot before it becomes coal. It's essentially oxygenated coal is what a Humate is. So it's just broken down plant matter that's been stable under the soil for millions of years, and we mine it out so that we can basically reapply that material. And that's the material that stores nutrients in every soil. So every soil on the planet needs humates to function and thrive and be able to hold and store nutrients. So we're just essentially, when we till, when we add high salt fertilizers, we're degrading that material. So it's a way to add it back in. And what's unique about this, when you think of humic, fulvic, everybody's probably heard of those or familiar with those terms. It's really the same as salt, silt, or sand, silt, clay. It's just the particle size that's the difference. So humic is a really big molecule. Fulvic's going to get a little smaller. And this is a material that's surface. It's mined on the surface, so it's not buried. So it's in the Pacific Northwest, but it's a lot smaller molecule size, so you need a lot less to work to be effective. And then it's also very easy to work with compared to, if you guys have used Humics, they're pretty messy, they're thick, viscous. But this is a way to solve a lot of those easy use issues. So you need, what 2 gallons of a Humic does, you need about a quart with this material to be able to accomplish the same thing. So that's a big benefit of this compared to other products out there. Then again, just the nutrient storage, water storage, soil structure. So something that every soil needs and can benefit from, both short and long term. That's a good, something to add in. And Habit P. So the next product here is one of our, say our flagship product. We did a lot of trials this year on this material. So what we do is we take that small humate, we add it in with the phosphorus. What that does is it binds to the phosphorus to keep it available. So phosphorus is anywhere from 60 to 80% inefficient. So it gets tied up with calcium, iron, clay, aluminum at different pHs. So it's a way to, just get the phosphorus you need to the plant as a starter without, without having the inefficiencies of those high salt traditional products. We had 95% success on 20 farms this year. And success defined as we compared it to a high phos, high salt starter of 5 gallons. We put 2 quarts to the acre of this down. You go 10 to 1 dilution, so it's a very highly diluted, low salt, so extremely low salt formula. Easy to work with, friendly with the biology, but we were able to range within a, plus or minus 3 bushels, so there wasn't any statistical significance within the trials, so we're very confident that we can perform, with those high salt starters. And it's something that instead of adding salt, degrading your soil, you're adding lower salt, getting the phos you need to start the plant, but then building up the soil, keeping the soil profile healthy is the main goal there. So that's a pretty exciting product. The one, if you had one product to try, this is the one we'd steer you towards, just to get a ton of value and be able to have the convenience, not have all the totes, logistics, all that kind of thing. So that's the Habit P. Next, we're going to get into Catalyst, I'm pretty sure. Yep. So Catalyst is you see the active ingredient is on the top there. It's all those lines. It's called a mass spectrometer reading. So that's essentially, it tells you the different molecule sizes within the formula. So there's over 400 or 500 different active ingredients, essentially carbs, sugars, enzymes, signaling molecules. There's a lot of diversity within that product, and it's really, there's a lot of sugar, molasses products. Those are simple sugars that are feeding mainly the bacteria. This is feeding a wider, more diverse set of microbes so that you can get that soil function improved. So you can see the 2 graphs there. You're increasing the diversity of the microbes and then also the activity, so the populations are going up too. So all the native nitrogen fixers, phos solubilizers, all those native microbes, you're just making those perform better, creating more of them. So you're mimicking a lot of what a cover crop does in a sense, and making nutrients more available. And then our foliars, Taurus is going to be our, it's a peptide is the active ingredient. So it's 35% plant-derived peptides is the active ingredient. So what is that? You're probably like, what the heck is that? So you can see all those different individual amino acids. That's called an aminogram. So that's essentially what the active ingredient of the product is. It's tough to get that on a label. They're working on all the tools to be able to do that. So if you look at the label, it doesn't tell you much about the actual active, unfortunately, due to the nature of how you have to register these products. But the peptide, if you have an amino acid, a chain of amino acids is a peptide, and then a chain of peptides is a protein. So a lot of the reason we add nitrogen or different minerals is to help synthesize amino acids within the plant. So this is a way just to add those amino acids to the plant, take the energy burden off the plant, and then those are going to trigger different stress, be able to tell the plant, hey, go acquire nutrients. It'll help metabolize chemicals a lot better, help mitigate chemical stress with any harsh chemistries. Then root growth, so it's going to have a ton of, we can go probably about half hour just into the benefits of peptides alone, so we won't go into that. Come talk to us after if you want more of the detail, but there's a ton of advantages to doing that. And then our Nourish Vitals is going to be when we mix the peptides with nutrients, it's going to make it more efficient delivery into the plant. So we're taking an NPK micro pack mix. And then, Dean, what are we looking at on the right with that chart there? I'll let the agronomist explain that.
Yeah, that's basically, nutrients, as you know, have a plus or minus charge. And when we add any particular nutrient, they can tie up other nutrients. We often think, well, more of a nutrient is better. But what can happen is if we throw too much of one nutrient on, we can create shortages in a bunch of the other nutrients. So Vitals is designed to be a balanced nutrient package, so it really does help stimulate the photosynthesis, make sure all the different nutrients are there. so that the plant can increase its photosynthesis and try to help to feed those microbes more and build the soil up further.
Yep, exactly. So the chart we had earlier with all the nutrient requirements, it's like, hey, let's tell the plant you have everything you need, especially right when you go into reproductive, then it can kick it into that reproductive mode and just try to get you what you need there. And then the peptide just allows safe delivery, so no risk, less risk of crop burn or anything like that, salt damage. So it makes it a lot friendlier to get into the crop. So here's just a complete picture of the whole. Just buzz through here. So that's just a picture of everything within the portfolio. We'll hand you out a little growing count. We'll get into how do we use these products or when, how do we actually adopt any of this if we want to try it. So pre-emerge herbicide, the catalyst to be the product you can use there. So you just tank mix that right in with it's not a living product, so you can mix it right in with your herbicides, and it's going to give you that soil boost. Then Habit P and Habit Boost. And Habit P is going to replace your starter. The Boost is going to be something you can add to an existing starter. If you're like, hey, I'm not buying on the Habit P, I'm good there, I want to use my starter I have. That's a way to at least make it work better, mitigate some of the salt damage, and just get better availability of your phos with that product. And then if you want to kick it up a notch, then the Inject N, the Myco, the Catalyst are things to add there. If you're using insecticide, would steer away from the Inject N. The Myco will work. There's a list of approved insecticides on the label on that one. If you're in super high, over 40 pounds of phos, I would steer away from the Myco, honestly. Just if you have too much phos, it's going to shut down your mycorrhizal growth. So you're better off going with something like an Habit P or an Habit Boost to start. And then once you get your phos levels, available phos down a little bit, then you can, the mycorrhizal will help to gain that. And the Catalyst is just something you can throw into anything, and it's going to be more resilient as far as what you mix it with there. Then at Sidrus, using your Habit Boost as a nutrient stabilizer, nutrient storage. And then your post-emerge pesticides, either an herbicide, fungicide, insecticide, the Taurus or the Vitals. If you're looking for more biostimulant effect or nutrient acquisition or using harsh chemistries, you want more of that biostimulant effect, then the Taurus is the one more to lean to. If you're looking for more of an NPK package, the Vitals is the one to use. And then if you want the best of both worlds, you can combine the two. There's a chart on the back that tells you how to combine the rates if you want to get the full benefit of the two products. So yeah, let's get into some proof of performance here. So this is what are we looking at with this boost here, Dean, on the left and the right?
Well, the control was on the left. This is the same plants, just two different views. And we did the boost on this product. You can see the much larger root mass, and a lot of that is when the nitrogen enters as an amino acid, and we get the control of phosphorus, we get a much bigger root mass. You can also see that it's dark, that black color. That only happens when we are getting those exudates and building, the carbon and organic matter levels in the soil. So when we dig this up, that's something we want to see, that we're getting this exudation and we're actually building the soil.
Yep, yeah, and something if you go do some side-by-sides or try it, like you can go dig. It's just pretty consistent. I went to a lot of fields, and this is pretty much just about any scenario. You can go dig the root mass, and it's pretty crazy. This is within 3 months we got this result on the physical color of the soil. So go see for yourself, and that's, instead of just driving by the field looking, does it look any greener, those types of things. It's really the soil test, and I always like to think if you could flip the field upside down, these products would be super easy to sell. But since you can't do that, it makes it more challenging. It's a lot of work to really go see, validate the performance and understand what they're doing. Okay. And then this is showing the Habit P. This is a trial in South Dakota. So you can see on the right, the root mass, the above ground growth is a lot more vigorous, but and you can see that excess phosphorus on the left is actually shutting down a lot of the mycorrhizal growth. So we didn't add any mycorrhizal product to this. Just by using less phosphorus, we're able to enable more root growth, which is going to go mine more phosphorus inherently. So you're really doing two things with the Habit P. You're giving it the phosphorus it needs to get started, but then you're also negating the phosphorus that's limiting the root growth below ground. And you can just like to envision, like when like a cover crop, you're always trying to, how do I get more living roots in the ground? That's a lot of what they preach there. So this is a way to really, you can see how you're making your cash crop perform some of the benefits you're trying to get with the cover crop by doing this stuff. This is showing, it's like, okay, well, I'm using that much less phos. There's got to be, like, it's not going to show up in the field, or it's going to be uneven. So this is the 1034 versus the Habit P. you can see across the field it's pretty indistinguishable. You're using a lot less phos, but you're not really noticing any, it's not set back or anything, just the varietal difference you can see there. But when you can see the starter shut off on the left-hand side there, then you can really, it's pretty prominent where it came up uneven, didn't come up at the same time. So when you take the starter out, you can really see where, the inhabit P is performing at par with that 1034. And then this is showing, we stacked a bunch of different products together in a field, and then the right is a competitive, one of the leading products out there, plus a 5 gallon per acre starter. So you can see we added a lot less phos, we added all the biology, and then you can see the soil difference on the left to the right. You're probably like, well, you just hit that with a hammer or something and broke it up. That's a bunch of balloon, but I'll show you the video here in the field in a second. And then this is a field in Minnesota where you can see the side-by-side where we basically filled in that dead area within the field. This is about 4 or 5 months. The washed out something, whether drainage or what it was, you can see where you can kind of find that outline in the left-hand side where it filled in that bad biology and we were able to actually grow a crop in that, and that was using a stacked, pretty loaded, kind of throwing everything we could at it. We weren't shooting for that. It just was one of the, something we noticed at this trial that was kind of cool. We'll close out. Here's a couple of videos. So this is that soil difference on the left. This is going to be with all those products. Goes in nice, digs up easy. You can just, flocculates apart. Watch I think I dig twice here.
Yeah, you can notice how the shovel goes in most of the way down.
Yep, so nice, good. Good structure, flocculates, falls apart, and then this was about probably 20 feet into the control. So this is with that 5 gallon.
Yeah, and jumping on the shovel with 2 feet and still didn't get in.
Yep, I've never been called dainty, so you can imagine that wasn't it was. But yeah, so those are the types of results. If you go dig and look at, it's a really good way to measure. So like we said, aside from just using yield as a metric, it's really using that soil health as, go dig, go look at roots. And then it's really about building that long-term quality. So you want to design the portfolio not to just be, hey, throw this on, hopefully I gain a bushel or two. We really want to give you tools. We're hoping that's the outcome, but really give you the tools to build that soil health in a good way and balance the negative effects of the fertilizer that we're, obviously we're not saying go quit your fertilizer. As we said, it's more, how do we just add the right things to be able to counteract some of the negatives over time. And then data tools. So as far as, we run some trials in-house that we do, but also if you guys, we have a lot of tools within our FBN membership. We have strip trial reports, data tools. We're like, hey, I'm skeptical. I need to go try this. I want to go dig, get soil tests, convince myself that what Dean and Neil are saying is there's something to it. So I know you can run strip trials on your own farm. We have a soil health protocol that we developed with Ward Labs we use for all of our trials. So if we included total nutrient storage, so to get that picture of, okay, well, most of the tests out there are designed to say, well, here's the available phos or the available N, but how do we actually get a picture of the total N? Because to get the picture the Dean was showing of the total phosphorus available. So we have a total nutrient test, it's called. It'll show you that, 200, 300 year supply versus just the available supply. So most soil tests, those are meant to sell you more fertilizer. Not to rag on them too much, but we just want to give you a total picture, unbiased picture of what's actually going on in the soil with that protocol.
Well, and another part of that is if there is a mineral short, the biology isn't going to be able to get it out of the soil. So it helps to indicate if you do really have a mineral shortage and should be adding something.
Yep. Yeah. So I'd say that as far as how to adopt these or, get to using them, it's, if you're convinced, you can obviously go all in. But if you're like, well, I just want to kind of figure this out and monitor results, really use these tools and then that protocol and go, don't just drive by the field, look at it. I'd say give it, 2 to 3 years on the same few fields to really see the cumulative results. Sometimes, the stuff doesn't always happen overnight or in 3-4 months. A lot of the benefits of these products, use them for 2, 3, 4 years, the soil benefits especially start to build up. So really encourage you guys to give it, 2 to 3 years, do the soil testing, go dig. If you just drive by and look at I've seen a lot of these trials, and it's a lot of times hard to just see it. It's something, it's probably, if you dig, it's usually pretty prominent, but over the top, it's always pretty hard to, even with the drone footage, you can sometimes find some results, but it's not always, the stark side-by-sides where there's like a 2 foot taller plant are usually pretty few and far in between. So anyway, that's the And then we have our IRL. So if you have interest, we do run trials on new products and some of the current products we have. So if you do have interest, come find us. RJ would be and Luke Samuel are the contacts there to be able to sign up for those. So something we're just trying to find new technologies, improve the portfolio, add to it. So really have a good vetting structure in place. So when we bring you guys to new products, that we're doing a lot of quality vetting beforehand to make sure they're working. So that's that. And we can go on to Q&A here.
Yep. So if anyone has a question, just raise your hand and they'll bring you a mic because we're recording. They won't be able to hear the question without the mic. And Neil and I are downstairs as well for the 3 days. So anybody that, if you have a more complicated question or shy, you don't want to ask it in the front of the group, you be sure to stop by our booth area and we can help you out there too.
I'm from West Tennessee. According to our PLFA test we pulled on some of our farms, Mycorizol is a lot lower than I expect, generally 0 to 2%. I've experimented with some other Mycorizol dry products, seed inoculants, and really haven't seen any results from the trials. I've tried other methods of reintroducing Mycorizol into our soils. The product you got, I think, has to be injected with like starter fertilizer or something. If we're not set up with that kind of system on our planter, do you have any other ways of introducing Mycorizol into our soils?
You basically seed treat it if you want it as well, like to put it right directly onto the seed.
That same product, or do you have a different product?
Same one. You can do a slurry seed treatment with that one.
And how does the fungicide seed treatments, how would it affect the Mycorrhiza product?
Yep, so we've done pretty testing to be able to verify if it affects colonization. But there's not a hard and fast rule, but we put a list on the back of the approved ones, so it is on the label which ones it'll work with, which ones there's issues. If there's
there's some that will definitely hinder it. So there's a list on the package there of what's usable with the mycorrhiza. And also to watch the phosphorus. If your phos levels are too high, that will prevent a compound called striglatitone, and you won't get mycorrhizal colonization on your roots.
Cool. Any other questions? Yeah.
We do have a handout too before everyone leaves if you're interested in just the timing charts.
Yeah, thanks everybody for joining us today. If you have any questions afterwards, again, feel free. We'll be around the show for the next 3 days, and have a good time at Farmer to Farmer, and hopefully catch you around, and appreciate your time.